I'm Daniel Dornbusch, the CEO of Excision, as the wonderful moderator, thank you, has talked about. We're going to tell you a little bit about what we're doing here in terms of clinical progress for the company in gene therapy. So at Excision, we talk about cutting-edge cures, and if you pardon the pun, of course we need to talk about disclosures because we will be talking about forward-looking statements today. We're developing CRISPR-based therapies to cure viral infectious diseases and improve the lives of chronically ill patients. So several aspects that you'll hear in the next couple of minutes that are quite different from what many other companies around are doing. First, talking about viral infectious diseases, there is some symmetry and perhaps poetry to this. We're using viruses to go kill viruses and eliminate them. We've been at this for quite some time, actually. Founded in 2015, we are headquartered in San Francisco. Our labs are in Cambridge, Massachusetts. And we've, of course, been working in a very large range of patents and IP. We've got exclusive licenses from both Jennifer Doudna's lab for the Cas12 family for viral infectious diseases, as well as Kamel Khalili's lab at Temple University for the approach to curing viral infectious diseases. Where these have come together is we are the first and only company to remove viral genomes from an animal and cure an animal of HIV. We're now working on a series of other programs, which we'll get to in just a minute as we progress these. And yes, we are now a clinical stage company. So we've announced just earlier this year that we've initiated our phase 1/2 trial in HIV. This is a single administration dose for potential cure for HIV. We'll tell you about that trial today. It's important, I think, to start with a couple of foundational explanations. First is, how are we doing what we're doing? Because if you've been in gene therapy for a while, you have heard the story that different groups around the world have attempted to cure viral infectious diseases without success. And the difference that we often talk about is the left and right here. So in the left, this is the traditional approach, whether it's zinc fingers, whether it's now CRISPR, but whether it was meganucleases in years past, all of the attempts have actually not succeeded in curing animals of HIV. You can do it quite well in vitro, in cell culture, but once it gets to an animal and a full population, it has not been successful. So what we're showing on the left is the single cut approach, and now this has been actually recapitulated by several labs around the world, that making a single cut in a viral genome is ineffective to deactivate it. The catch and the innovation that we've brought to this, and actually facilitated by CRISPR's discovery, is making more than one cut. Because by making more than one cut, we can actually eliminate a large section of viral DNA and achieve a cure. We've created a stylized, so excuse the stylization of stretching out DNA, but we do have a video to show when you look at, and what we're trying to show here is normal DNA being the light, the white DNA, the viral DNA being the dark gray. What we're doing is introducing two CRISPR proteins and two guides that target, importantly, viral DNA, not human DNA, make their excisions and remove large sections, we're talking about thousands of base pairs of viral DNA, to eliminate any chance of reproduction. This allows what we describe as three essential elements to facilitate this as an actual therapeutic in patients. The first is efficacy, gene targeting and cutting. Where others' approaches modify human genome sequences, what we're looking at is we're targeting non-human DNA. So in a safety and an efficacy pair here, we can go after and select. In fact, I'll show you more detail on the next slide about how we can very specifically select non-human DNA to ensure the absolute minimal of off-target potential that we've published actually earlier this year. Second is the gene editing effect. We're not inserting anything new into host genomes, which could potentially disrupt function. And third is the duration of efficacy. We're not trying to establish a constitutive protein expression for the lifetime of a patient. All these therapies need to do is go in, remove viral DNA, and be done. How are we doing what we're doing? We're using actually a range of different nucleases. This is an explanation of the next generation of CRISPR editors. You may have heard this story with Scribe or with Mammoth. We have an exclusive license from Berkeley for viral infectious diseases. Importantly, in the Cas12 family, and that's what we're showing here, there's some elements to this family that lend itself to efficacy in what we're doing in our approach, meaning removal of DNA, it's a higher increase of MMEJ versus NHEJ, and what we're talking about is the four key benefits on the right. Smaller size improves packaging. The generation of overhangs includes that and leads to a higher chance of excision. 3, additional PAM sites, and 4, reduced chance of pre-existing immunity. What we've been building over the years, and yes, we've actually been at this for almost a decade now, is we talk about a 6-part or describe a 6-part approach to this. This is all our bioinformatics approach about how we build the therapeutics that we build, starting with viral database curations, moving on to consensus and genome building, conserved gRNA identifications, cross-referencing against the human genome. So what we've built in the first 4 is actually customized approaches, patented approaches, and actually customized software in order to build what we're building. Our chief scientific officer often loves to say that he's done this in years past at different companies, at Sangamo and others, and it requires a concerted, focused effort exclusively in viral infectious diseases to build what we're building here. To highlight one thing on this slide is number 5. What we're able to do, because we're targeting non-human DNA, is we can select in the first 4 components here the targets in viral DNA and then cross-reference it against anything else in the human genome, thereby maximizing the chance for an on-target cut and minimizing the chance for an off. What this has built is a pipeline so far of 4 programs. The first is in the clinic, and we'll talk about that in just a minute. This is EBT-101 for HIV, EBT-104 for herpes virus, followed by EBT-103 for PML, which is, of course, the virus that causes, JC virus, which causes PML, and then EBT-107 for hepatitis B. Each of these indications at the moment are designed to be one-and-dones. We're using AAV9 in the first program, and EBT-101, as well as for EBT-103 for JC virus. For herpes virus and Hep B, it allows us to target other indications, of course, Hep B being targeted in the liver. It's not too far for the rest of this this esteemed group of gene editing therapists to know what we're probably targeting with Hep B. It's probably worth mentioning one thing. Most of this group may not be viral infectious disease experts. Usually when we talk to viral infectious disease, they look at this list and say, this is really impressive. This is the first time we know this data because it's the first time HIV has ever been cured in an animal. When we usually talk to gene editing experts, they usually look at these and say, how can you possibly be going after these? Don't you need to get every virus in every cell? And the answer is actually no. We could spend the next couple of hours going through all the literature on this, but the point is that there's actually been quite a font of published publications. Not ours, these are others in the field, as well as multiple clinical trials showing how individuals do not need to get rid of every single cell. And that we're looking at percentages of these in order to achieve a cure. If you want to learn more, please visit our website. There's quite a few publications on this. Another differentiating factor that we are focused on quite acutely is the size of the markets we're going after. HIV individually, we're looking at standards of care and incidence and prevalence here. 1 million cases in the US, 1.2 million in Europe, 38 million worldwide. JC virus and PML is the one orphan disease of this pipeline. Only about 4,000 individuals in the US and Europe contract PML or are diagnosed with PML per year. But once we get to herpes virus and hepatitis B, and we noted one sub-indication in herpes virus here, the populations are so large, millions, hundreds of millions, and in case of herpes virus, actually billions of patients. What it affords us is to target some of the subsets of these indications for the, as some say, the worst patients of the worst, so that those that suffer the most, we can target those. Happy to jump into EBT-101 with our clinical trial. So talking about HIV, of course, yes, the care has transformed in the last several years with antiretroviral therapy. Patients are living relatively normal, healthy lives and not dying nearly at the rate they were decades ago. However, the care is complex and it's costly. What is not advertised all that well, when you look at it's now advertised on, on television advertisements, is the level of and the number of side effects these individuals, people living with HIV, have. Not necessarily only according to their antiretrovirals. It has to do with real-world evidence. When you start stacking antiretrovirals on top of anything from immune disorder therapeutics, antidepressants, the statistics on side effects for HIV, and these are well published, more than 50% have peripheral neuropathy, more than 50% have metabolic disorders, more than 60% have levels of dementia and neuronal issues. The last is costly. Published, fortunately, we don't have to go do these studies ourselves. There's been multiple publications on the cost of people living with HIV, incremental to those who do not have HIV. And as we mentioned here in one publication, it's over $1.5 million for these patients. I mentioned we've been at this for a while. We've got a whole host. I'm only going to show one preclinical slide here, but we've got plenty of publications. You see the bullet points on the left. If you'd like to delve into the preclinical, please, we can have those discussions offline. But what we've walked through over the years is in vitro, humanized mouse, and then primate studies. We've now completed 2 primate studies in HIV as we've moved forward to the clinic. We've submitted this to the FDA. We've announced that we got clearance from the FDA for this phase 1/2 clinical trial. We've initiated it this year and dosed our first patient, as we just announced a couple of weeks ago. So this is a phase 1/2 study, meaning safety, both safety and efficacy. We're looking at 18 to 60-year-old males and females and these are people living with HIV. The clinical data is so strong and so extensive with people living with HIV, we do not need to do, or as the FDA has advised, we do not need to do a control arm with this. We know what happens. There's been plenty of clinical trials testing what happens with patients when they stop their antiretrovirals. We know that 50% rebound within 3.5 weeks, and just about everybody rebounds within 8 weeks after stopping therapy. And as you see in the endpoints in the bottom here, our primary endpoint, of course, is safety, as this is a first-in-human trial, but also biodistribution, excision assessment, meaning are we cutting HIV for the first time ever in a human? Reservoir assessment, are we reducing the HIV reservoir? And of course, the last one is delayed rebound. So built into this, as you can tell on clinicaltrials.gov, built into this trial is people will go on to get a single 1-hour IV infusion. They will stay on their antiretrovirals. This is in green for 3 months. We're calling this a safety and efficacy assessment. And then right in the middle, the participant stops their antiretroviral therapy, and then we will look and assess for potential rebound or ideally lack of rebound. In the last minute, tell you about what we're building. It's just an outstanding group of individuals, both viral infectious disease experts, gene therapy experts, and of course biotechnology and commercialization experts. Between the R&D, the regulatory, the clinical, it's just an outstanding group of individuals. And a wonderful scientific advisory board similarly. So we get to balance quite a few different pieces, both the gene therapy, the gene editing, the delivery, as you'll see, and as well as the viral infectious disease experts to bring together really a novel combination of experts to help guide us through. And I'll just end with one quote, and this is Bob Gallo, the co-discoverer of HIV. He wrote an editorial just a few years ago talking about, and of course, he's been looking and working on potential cures for HIV for 40 years. He called this one the potential for a future bona fide cure for HIV. If Bob happens to be in the audience, thank you, Bob. We completely agree, and thank you for your time today.